Our topic for today is periodontal pathogenesis. A quick recap from yesterday is we had the healthy gingiva in which there is junctional epithelium. The junctional epithelium is oral epithelium plus reduced enamel epithelium. It is a collar-like band of stratified squamous epithelium. It is non-keratinizing and it forms the floor of the sulcus, which you can see here. Then we have the sulcular epithelium, which lines the gingival sulcus, is non-keratinized and it undergoes hydropic change. It functions as a semi-permeable membrane. This over here is the gingival margin, and this is the marginal gingiva. Over here we have the attached gingiva. The main antibodies included in healthy gingival response is IgA, which is present in the saliva, and IgG, which is the predominant immunoglobulin present in gingival crevicular fluid. Then we have gingivitis. Gingivitis is basically the inflammation of gingival tissues that does not involve PDL or alveolar bone. If it does, that would progress to periodontitis. There are 4 stages in gingivitis. We have the initial lesion, the early lesion, the established lesion, and the advanced lesion. The initial lesion takes 2 to 4 days of plaque accumulation for it to develop. The predominant cell type are neutrophils. There is increased gingival crevicular fluid flow and mild inflammation. Clinically, this appears as normal gingival tissue, and it does not bleed on probing yet. Then we have the early lesion. The early lesion takes 1 week of plaque accumulation. The predominant cell type is neutrophils plus lymphocytes, T lymphocyte cells to be specific. You will find vasodilation, increased gingival crevicular flow, and vascular permeability here. Clinically, it appears as early gingivitis, and there will be bleeding on probing in this case. The next one we have is the established lesion. The established lesion takes 2 to 3 weeks it develops 2 to 3 weeks after plaque accumulation. It is still reversible if it's established lesion. Once it turns to advanced lesion, it is not reversible anymore. Clinically, it appears as chronic gingivitis. The predominant cells are plasma cells and lymphocytes. There is bleeding on probing, and the important clinical sign over here is that there is gingival anoxemia, which clinically, it will look like a bluish hue present on your dark red gingiva, inflamed gingiva. We have the advanced lesion next. The advanced lesion, clinically, it leads to periodontitis. Because it leads to periodontitis, it is irreversible after this stage. There are 2 main cells. The first one is plasma cells, which are present in the connective tissue, and in the pocket epithelium, you will still have neutrophils present. What's happening here is that there is, as we discussed yesterday, we had passive eruption, active eruption. So once you have an advanced lesion, you have a sort of passive eruption because there's apical migration of the junctional epithelium. This is happening to prevent further entry of bacteria into the subgingival spaces. In this case, the pocket deepens. There will be a deepening of the pocket because of the apical migration of junctional epithelium. We have the inflammatory responses in the periodontium next. The inflammation responses can be microbial or it can be due to the host. So the microbial virulence factors, you just need to know the names of these. They're not really that important to remember. I haven't seen any BCQ from here as yet. The host-derived inflammatory mediators are actually important, and you have 3 main responses here by the cytokines, matrix metalloproteins, and interleukins. In the host-derived inflammatory mediators, basically the majority of tissue damage is due to the host immunity by dysregulated production of inflammatory mediators. So when you have an inflammatory reaction, there will be a lot of inflammatory mediators will be produced, and because of the dysregulated production, this is what leads to tissue damage. What are the mediators? We have cytokines. Cytokines, these signal, broadcast, and amplify the immune responses. These mediate bone destruction through induction of fibroblasts and osteoclasts. The fibroblasts and osteoclasts then produce matrix metalloproteinases, which are proteolytic enzymes. These break down the connective tissue and hence lead to destruction of bone. Matrix metalloproteinases. What are these? These are enzymes that degrade the extracellular matrix. The major matrix metalloproteinases present in periodontitis are MMP8 and MMP9, whereas normally you have MMP1 that predominates. We have interleukins next. The interleukins, what I found to be important here are only these 2 points, that interleukin 1 alpha, it mediates bone resorption, and interleukin 1 beta, its main function is adaptive immunity. The resolution of inflammation is by lipoxins, resolvins, and protectins. Then we have gingival crevicular fluid. It originates from the postcapillary venules of gingival plexus, and it has 2 functions. One is to clean the gingival crevice by flushing action. The next function is to bring neutrophils and other blood components to the site when there is inflammation. The gingival crevicular fluid flow will increase in case of inflammation, but in normal circumstances, it is also present. Neutrophils are always present, whether you have a healthy gingiva or you have an inflamed gingiva, neutrophils will always be present, and these migrate through junctional epithelium into the sulcus when there is inflammation. We have adaptive immunity next. Adaptive immunity is mediated by lymphocytes. Now you have T lymphocytes and you have B lymphocytes. In periodontitis, you have B lymphocytes. In gingivitis, we have T lymphocytes. T lymphocytes are CD4 T lymphocytes that predominate. CD4 T lymphocytes can be divided into Th1 and Th2. Th1 is interferon gamma. The interferon gamma, it contains macrophages, NK cells, CD8 cytotoxic T cells. The macrophages, these phagocytize and kill microbial pathogens, whereas the rest of them, NK cells and CD8 cytotoxic cells, T cells, these kill infected host cells. So macrophages kill microbial pathogens, but NK cells and CD8, these kill host cells. Then we have Th2, which is, it progresses as humoral immunity and is the B cell response. This contains interleukin 4, 5, and 13. Now the Th2 response, it leads to a B cell response, and as we discussed previously, B cell response is in periodontitis. Henceforth, this is a destructive lesion. The Th1 response is it is not a B cell response, it is a T cell response, and henceforth is a stable periodontal lesion. That wraps up our chapter of periodontal pathogenesis. I hope you guys understood.